With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
Hello students, the question here is about the packaging of the DNA helix. So packaging of DNA helix is an important thing because if we talk about eukaryotes have 6.6 into 10 raised to power 9 base pairs, right? So they are 10 raised to power 9 base pairs. In a haploid content, there are 3.3 into 10 raised to power 9 base pairs. So these many number of base pairs, if we take the distance between each base pairs as 0.34 nanometer and we multiply it with to convert it into meters. So when we find it out that the distance between them is 0.34 into 10 raised to power minus 9 meters, and we try to find out the length of the DNA, a length from the single cell of the DNA, the DNA length will be approximately 2.2 meters. So you can imagine such a small cell which cannot be even seen with the naked eye, and the nucleus will be even smaller than that. So in that there is packaging of the DNA. So it has to be very, very intricately packed so that it can stay there in the nucleus in a very, very compact form. So the packaging of DNA in the eukaryotes, specifically talking about the eukaryotes, takes place with the help of the major proteins are the histone proteins. These are the basic proteins which are rich in basic amino acids such as lysine and arginine, and 8 of these histone proteins will form a histone octamer around which 166 base pair length of DNA is wrapped around to form a structure which is called as nucleosome. So these nucleosome will be further coiled up, and nucleosome gets coiled up to form solenoid, then the chromatin fiber, then the chromatid, and then the chromosome. This chromosome has got approximately 1400 nanometer diameter, whereas there are other set of proteins which are called as non-histone chromosomal proteins, which are no doubt not the part of the nucleosome, but they help in the additional packing of the DNA in case of eukaryotes. While prokaryotes do not have histone proteins. So in prokaryotes, this is one reason why the DNA in prokaryotes is called as a naked DNA. So one reason is because of the absence of the histone proteins, while how will the DNA be packed in prokaryotes is with the help of non-histone basic proteins like polyamines. Right? So polyamines are involved in packaging of DNA in prokaryotes. So let's see the options which are given here. The first option says it involves polyamines in eukaryotes. This is now certainly longer. We have already seen the explanation. The second is occurs with the help of NHC proteins only. No, it requires histone proteins also along with this. So this is also wrong. Requires acidic proteins that help in coiling of DNA in prokaryotes. No, these are the basic proteins. In fact, polyamines are the basic proteins. So this is also wrong. Then it says that it is more complex in eukaryotes than in prokaryotes. Yes, this packaging is much more complex in case of eukaryotes involving the histone and the non-histone chromosomal proteins which are coiled to various levels, right? So the option number 4 here becomes the right answer for this question. This is all about this query. Thank you.
For class 6 to 12, IIT JEE and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.